Started. So I have to record the lectures. So this is now. I'm going to start giving you a quick presentation. So let me share my screen here. Okay, can everybody see my screen? Good. Okay, so as I mentioned before, my name is Lauren Dupuis, and I'm just going to give you a very basic introduction to what we're going to be doing in this session. So one thing is I'm going to use Slido for some interaction with you guys, just to ask a couple of questions. So if you go to slido.com and you use that code 169659, then it should open up for when I answer or ask you the questions. So just so everybody has this open as well. And if you want to wait, you can also use it with a QR code on your phone if you prefer. So when the questions come up, then there should be a QR code next to it that you can scan. Okay, so I'll start by introducing briefly our instructors. So of course we've kind of already introduced ourselves as well, but then we have, oh, how come this is not working? Yeah, okay, so first we have John Scooter Morris. He's, as he said, the executive director, a resource on biocomputing, visualization, and informatics at UCSF. And also I found out a roving engineer, I think he called it. And then we have, good. Then we have Martina Summer Kutmon, who is an assistant professor at the Department of Bioinformatics and Maastricht Center for Systems Biology here at Maastricht University. And myself, Lauren Dupuis, I'm a postdoc at the Department of Bioinformatics at Maastricht University. So what are we going to get into today? So basically, as everybody is all too aware of this whole COVID situation, we are going to kind of base our tutorial around this COVID data set. And so we're going to be focusing really on things that were developed with the COVID-19 disease maps project and the Wiki Pathways team, as well as the Cytoscape team. So of course, we've split this down into 2 days. And in the first day, we're really going to be all about the tools. So we're going to start with an introduction to the COVID-19 disease maps project and the Wiki Pathways by Martina. And then Scooter is going to continue by introducing Cytoscape, the tool. And he will continue with some introduction into how to using automation in Cytoscape with R. And so today we'll really be focusing on understanding the tools. And tomorrow we're going to take our toolbox and it's going to be all about the application. And the point is to kind of try to understand these molecular mechanisms underlying the COVID-19 pandemic situation. And so we're going to take this toolbox and we're going to apply it with a COVID-19 data set. And so the more detailed program, what we're going to get into today, I think we're already a minute behind, but so I'll give the introduction briefly. As you see, then I think all of you have access to this program, but next will be an introduction to the COVID-19 disease maps, Wiki Pathways, an introduction to Cytoscape, some hands-on, and some wrap-up. And then for tomorrow, it'll be really applying these tools and to different workflows. So we'll have 3 different workflows for you to go through.
Welcome, everyone. I hope you can see my screen. I'm now going to give an introduction about the resources that you'll mainly use tomorrow. And that's for the really molecular mechanisms related to COVID-19 that will give us a little bit of a basic of understanding of what's actually going on. Just a quick start. Let me see. Yeah. I'm also going to use Slido for 2 quick questions, but you can also type it in the chat if you haven't made the connection yet. It's a different code this time, so 352618. And what I wanted to mention here as well is if you don't want to wait with your question to write it down until the end, you can also put them into the Slido app through the whole presentation, and then they will show up at the end. So don't wait until the end. I have two polls at the start because some of you mentioned it already. I know some of you were involved in the disease map project and so on. So I was interested in, now this doesn't look right. Wait a second. Sorry. This should not look like this. Okay, we're going to do this without the Slido parts, but sorry about that. You can test it as often as you want. But yeah, my idea was to ask how many of you know about the COVID-19 disease map and how many of you know about Wiki Pathways, but feel free to ask any questions at the end. Sorry about that. Now, we all know that these molecules that we often measure and study, they don't function in isolation, but they all interact all the time, and they form these complex networks that define the cellular system. And when we measure this, it's usually measured as isolated data points. So we have a transcript measured, a protein measured, protein abundance measured, metabolites measured, and so on. And what we then try to do is we try to make comparative statistics to make sense of what these molecules are doing. And we try to cluster them. We try to find functional groups within them to kind of make sense of them. And with pathways and networks, we actually try to go a little bit further. We try to look at this functional organization of how these molecules are actually working and participating within a certain process. And that's what the whole part about Wiki Pathways and the disease map project is about. So these pathway models are really detailed curation of biological processes, but they're more than images. They're really machine readable pathway models that allows us to do intuitive data visualization, but they can also be used for downstream analysis in networks or computational modeling when we want to go further into different kinds of modeling strategies. And especially in situations like COVID, these are very important to better understand, but also validate and simulate molecular mechanisms. In COVID-19, we had information coming in every day. Things changed nearly on a daily basis in the beginning. So there was really an urgent need to kind of curate this information in a format that we can share and test if the situation actually works like that. There's different kind of biological pathways, but I wanted to use this example to show you the advantage of making a pathway model versus having a diagram. So on the left side, this is a static image published in 2016. It's a nice signaling pathway drawn, and the data is actually added with these little arrows next to the molecules. So you see maybe two red arrows up or a green arrow down. And it says tells us something about the level of increase or decrease in mRNA levels, protein levels, or phosphorylation states. So they had three different types of omics data. Here we have phosphorylation states, we have M for mRNA and P for protein. And a colleague of mine basically took this pathway image, which was a static image in a paper, and made a pathway model out of it, which allowed us to then load the data set in a quick way in different tools to visualize the data. In this case, we split the box in half. Left half is the mRNA, right half is the protein abundances, and also the phosphorylation states have different colors. So if we look at, for example, the PIK3CA in the top left, we see that the phosphorylation site is slightly upregulated. The mRNA level is slightly downregulated with a light blue color, and the protein is slightly upregulated with a light red. So we can now do this automatically, this visualization, and we can also load another data set immediately without redrawing the pathway figure in every single aspect. So making these machine readable allows us to do a lot more with them. And this is just visualization, but we can do even more than that. And this is also exactly what we did in the COVID-19 disease map project. So since 2020, there are over 140,000 publications about COVID-19. And this is just a PubMed query. I'm sure there's a lot more preprints and other stuff that doesn't end up in PubMed. So it's a huge amount of publications that turned up. And the molecular pathophysiology the virus infection is really complex. So there's many biological pathways involved with different cell types and organs that are affected. So it's a really systemic response. And so what the disease map project started was a collaborative effort. So it was I've been in the pathway field for a long time, and we had these smaller collaborations with different databases together and so on. This was the first time where we got really a huge group of researchers together, and they collaboratively worked on this disease map. So we had biocurators, we had domain experts, we had modelers, we had data analysts. So from all kinds of fields, text mining, everything came together and kind of really participated in establishing this resource and the communities with it. So we still have the weekly telcons where we give presentations to each other, where we exchange, where we discuss curation statuses and so on. And so the whole disease maps community, the whole Reactome database, Wiki Pathways, and so on, many other resources came together and kind of build up that community. And the aim was to have an open access collection of curated computational diagrams and models of the molecular mechanisms that are implicated in the disease, to be able to then help better understand the disease, get better treatments, and so on. So all the downstream things that we want to do. What you see here is actually, I'm not sure it's still complete. This was an early version of all the different tools and communities and things that got involved. So we have here Minerva in the center, where it's kind of integrating a lot of things, and the diagrams are in there, but the data comes from Reactome, from Wiki Pathways, from Cell Designer, from SPGNED, all kinds of formats involved, a lot of standardization that we try to do, but we don't focus on one, we try to support many different ones. There's a whole area on text mining, Cytoscape integration, modeling integration, then downstream really doing more Boolean simulations and so on. So the idea is really, we had all these different people, and let's get them all on board to kind of use this resource in its best way. This is the disease map, how it is organized right now. So we have this beautiful top layer view where we have all the different parts that make sense, that are part of it. And we had a kind of artistic person, an artist helping with the design of this. And then each of these sections actually has pathway models. So not all of the sections actually do have pathway models yet. They're still building up. Obviously, we're not there yet, but each of these, for example, for attachment and entry, there are several pathway models describing the different processes relevant for that specific section. If you go to the COVID-19 map for elixirluxembourg.org and /Minerva, you can actually interact with the map and click on them and see how it works. But it's a complex mechanisms and it's not complete yet, and we're still working on it. And a lot of people are involved in the process. If you then click on them on the diagrams, then you see the typical pathway models. In this case, it's SPGN visualizations of the model, where you see how the virus proteins, which are always here in pinkish, interact with the host processes, and what the response might be of the host cell. Currently, we have 41 diagrams in the disease map. Some of them are in the Minerva platform. Some of them are linked from the Minerva platform to other resources. We have 1,836 interactions between 5,499 elements. There's a lot of complexes in there and so on. And we also make sure that all of these maps are really strongly supported by publications and sometimes preprints. So there's over 600 publications that are referenced in our pathway maps. The nice thing about this integration is that the models are automatically available in SBML, SPGNML, and GPML, so you can use them for the different types of analysis. And the conversions and integration is then supported by Minerva. Now, one of the resources that is integrated in the disease map, and that's also what I'm personally involved in a lot, is the Wiki Pathways project. So I'm an architect since 2012. It was launched in 2008 as an experiment. So it was kind of, we have these manually curated databases, but the amount of knowledge that's being published is so large and it's so fast that it's difficult to keep these up to date. So the idea of Wiki Pathways was, can we take advantage of the participation of communities to to contribute this content directly. So this is the idea of crowdsourcing. And since then, Wiki Pathways has grown immensely, and we support a lot of different species and communities on the database. So Wiki Pathways is like a Wikipedia for pathways. So it's built on MediaWiki, but the main idea is really this collection and curation of knowledge from the community for the community. So everybody can contribute, discuss, edit, curate, and use the pathway collections. And we also collaborate with a lot of different databases. The advantages of a platform like that is that the new findings can be added immediately. So the COVID-19, we had the first portal up in, I think, March 2020, we had the portal up, and the first pathways were submitted in late March or early April 2020. So you can immediately put it up and discuss things. It's collaborative, so we really support the exchange of ideas and discuss the pathways, and we collaborate with other manually curated pathway databases like Reactome or NetPath. The other idea that we have in Wiki Pathways is that we want to keep the platform quite flexible. So we allow people to submit pathways that are still under development or hypothetical pathways, as well as disease-specific or cell type-specific pathways. All of these tags are then actually, or all of these categories are tagged. So it's not that every pathway automatically ends up in an analysis collection, but we have assessment of the quality in the database. So on a pathway page, you have the title and the authors. The authors are always listed. The first author is always first. And then the contributors are ordered by the amount and edits made. The diagram is then clickable and browsable, zoomable, and so on. So you get information about the data nodes and so on. And below the pathway, you can find quality tags. And these quality tags or collection tags, they give you an indication about the status of the pathway. So for example, if it's a hypothetical pathway, or if it's still under development, then it is not in an analysis collection yet. So it's not in the curated collection that you have available for download. Every pathway can have ontology tags. So we have the pathway ontology supported, but we also want to indicate if it's specific for a disease or specific for a cell type, because that might be interesting for some users. There's a bibliography, so every element in the pathway can be annotated with literature references, and those are listed then for the pathway model as well. And the nice thing about a wiki system is that there's a history. So you can easily go back and revert to a previous version if needed. So it's really collaborative, and we have over 700 contributors to the whole content of the database. We do have a lot of external references that we support. So if you click on one of the elements in the pathway, is it a metabolite or a gene, you get link outs to other databases that might be important, have important information. I want to highlight the discussion page. So in the top left, you also see this. When you're logged in, you can start a discussion. So we always encourage also people having discussions about the pathway if there's questions or discussions about what should go in or not. And you can download the database, the pathways in different formats. I saw a question in the chat. So the question was about Keg Pathways. So we used to have a Keg Pathway converter, but since the license change, it's not possible to integrate Keg into Wiki Pathways. So the best way would probably be through Cytoscape, the integration, because there's a nice Cytoscape app if Keg Pathways is something you want to include, or you do it, the enrichment analysis in R, as we will do tomorrow, for example. But due to the license, so Wiki Pathways is completely CC0, so we're not allowed to integrate Keg Pathways. Something that I mentioned a little bit, but something that has really been pushed in the last 2-3 years is the concept of community portals on Wiki Pathways. So these are special interest groups that maintain this portal themselves. So there's a couple of people involved for each portal, and they highlight pathways relevant for their community. And you see we have a lot of different communities, from plants to COVID to rare diseases to cancer and so on. And they're still growing, so we still get requests regularly. So this is a quite nice way to get the communities involved as well. Currently, we have nearly 3,000 pathways from over 700 contributors. And just as an example, only 2,078 pathways are included in this curated collection. So there's still nearly 900 pathways that are under development or hypothetical or still need work. The focus in Wiki Pathways is still on human pathways, but we have converters so homology converters available if needed. And in the last months, as an example, there were 368 edits from 26 different contributors and 10 new pathways were added. So the content is steadily increasing. You can download the data per pathway, or you can download the collections as a whole. And there's different formats from TPML to image formats to other standards, biopacks, or now through Minerva, you can also download them as SBML or SBGN. If you're doing more programmatic access, we have different ways to do that as well through the REST API, the RDF, our Wiki Pathways. So these are all, we have the different kind of platforms that allow you. Our Wiki Pathways is something we'll use tomorrow as well. You can open them in Cytoscape with the Cytoscape app. That's also something we'll do. They are available in Index, all the human pathways, and in Wikidata. So they're also disseminated in all the other platforms that might be relevant. Now, a little bit in summary, what we really want to focus on is the COVID-19 molecular mechanisms. So we're really going to look at some of these pathways and what's happening with them in a COVID-specific data set. There's a lot of curation effort going on in the COVID-19 disease map project. If that's something you're really interested or you're missing something in that disease map, then please let us know, join the community, or give us a hint on where we need to look and what we're still missing. And so something we'll focus on tomorrow is how can we use these models now that we have available when we want to analyze our omics data. So we will focus, you will do today a little bit of a combination of user interface and learning how to do this programmatically with Scooter. And tomorrow it will all be about reproducible data analysis workflows. So it will be automated, and some parts are there that you just need to interpret, and some parts you will need to slightly adapt and try to include what you learned today. And we want to integrate the different resources in one type of analysis. Are there any questions at this point? You can either just unmute, raise a hand, or write them in the chat. Looks like it's all clear. So today you will really go into how can we use Cytoscape, how can we link the automated part to Cytoscape, already some examples with RNA-seq data sets and so on. And tomorrow we'll play through the whole COVID situation with 3 different applied workflows where we integrate the COVID-19 disease map and the Wiki Pathways COVID portal pathways in one type of analysis.
Can I ask one question, please?
Sure.
Yes, thank you very much. So I have a specific question, like first, what kind of omic we can use? And the second one is what kind of question we can like answer, like, yeah. I mean, question related to COVID-19, I think.
Yeah. So what we're going to do tomorrow is using a gene expression data set, because that's the most complete kind of visualization we can do. I've also analyzed proteomics data sets integrated from different tissues. So comparing different tissue proteomics data sets with the disease map. There's obviously also some metabolites in there. So you can integrate proteomics with metabolomics or gene expression as well. And a lot of people are using them for all kinds of data analysis in theory. For the enrichment analysis, you always need to be careful with proteomics data and more limited data, because the background, you might not know enough about that, but then really focus on visualization and network analysis approaches. So what we'll do in the workflow tomorrow is really focus on transcriptomics data or proteomics data. We do extend it with drug target information, but that's something where we don't have visualization for the metabolites per se in this case, but we can visualize the data of the metabolomics data in there as well. Is there specific omics data that you were wondering about that you wanted to have analyzed or integrated?
No, it's just proteomics, metabolomics, and transcriptomics. And are
Yeah.
all of these data comes from COVID patient?
So in terms of data availability, we now use a publicly available data set, right? So there's a lot of gene expression data available now. There's also quite some big proteomic studies that's being done. I haven't seen that many metabolomics data sets yet, but that's coming up as well. So in the different, I can show some papers and data sets later, or prepare for tomorrow a couple of slides or a spreadsheet where you can find some of the nice data sets that we use to analyze it as well.
No. My question: are these omics data profiled from the same patient, like the proteomics, the metabolomics, and transcriptomics? All of them, they came from the same patient?
For some data sets, yes, not for all data sets. Depends on the data set. So I'm more on the computational side. I do collaborate with different groups. It really depends on the project and the interest of the group that's measuring the data. So the most data is really still either genetics and transcriptomics. That's a combination that happens often. Proteomics, there's more focused data sets for proteomics, and as mentioned, metabolomics, I don't have. There's some examples that I know of, but not too many yet. And I see that there are some examples about focusing on proteomics and metabolomics. Yeah, I will make sure to highlight where the proteomics would fit in tomorrow as well. In the end, the identifier mapping will happen there anyway. So that a lot of the molecules are actually proteins in the pathway. So there is ID mapping happening automatically. Okay, in terms of time, I'm gonna hand over to Scooter, who will start his first introduction into Cytoscape.
Hi, everyone. We have a mix of expertise in Cytoscape here. So I'm going to skip through some things and go into depth in others. If you look at this slide set, it covers a substantial amount of content and way too much content for the time that we have, but that's quite intentional. So I think the idea as we're going through this is I want to provide the slides to you. You can peruse these slides and go through them in any kind of detail that you want. I'm going to focus on a couple of different things. I'm going to focus on a little bit about how you get data into Cytoscape. I'm going to focus on a little bit of background so that we're all at the same level about looking at networks. And I'm going to focus a little bit on data visualization in this section. Then we're going to have a sort of a long hands-on section where I've got some transcriptomic data that everybody can explore and actually develop networks using the transcriptomic data that we're going to provide and string. Hopefully that's going to meet, everybody's needs. The goal here is everybody should be able to get something out of this, even if you're an expert. If not, ask questions. I'm not going to use Slido or anything. We're a really small group. So what I'd love to have people do, it'll help me stay awake, is ask questions, either in the chat or just go ahead and interrupt me. I think I'm really interested in getting feedback, and that'll help guide this. As I said, there's more content here than we can go through in the time that we have. Okay, this is the rough outline. I'm going to talk a little bit about network biology to get us level set. I'm going to talk about how you go about finding network data. Tina already talked a little bit about pathways, and we'll talk a little bit more about those and put that in context with other kinds of network data. Then we're going to talk very briefly about network analysis. I'm going to literally boil all network analysis down to one slide. But mostly we'll talk about visualization. From there, we'll talk about Cytoscape. and I'll give a brief introduction to Cytoscape, and then we'll dive into the hands-on exercises. Does that work for everybody? Sort of open season here. Does this seem about right? Hands up or whatever seems to be good for you. Okay, great, great. All those who aren't hands up must be the experts in Cytoscape and they're going and getting coffee or whatever. That's fine. All right, so we'll start here. Okay, so introduction to network biology. I've already mentioned this. So networks. Why do we care so much about networks? I mean, Cytoscape has been around now for almost 20 years, and it has been incredibly popular because of the network orientation. So the immediate question is why? The first answer is networks are similar, they're just everywhere, especially in biology. When you think about it, most of us who went through biochemistry and learned the Krebs cycle, that's a network. So you see molecular networks, you see networks in cell-cell communication, nervous systems are often structured as networks, and even now social networks are in fact networks. But also they're very powerful tools. We can reduce complexity by using networks. And I'll show you a slide with some collaborators that really bring that home. They're more efficient than tables, and they're really great for data integration. I can add additional channels of information to networks relatively easily. And they do give us an intuitive visualization if they're done right. So I'm going to take a transcriptomics, a brief tour through the history of transcriptomics visualizations. We started this way. This is what transcriptomics looked like. And then we got microarrays, and this is how we looked at microarray data. And then we got down to clustering our microarray data. I'm going to take a brief segue here. We don't do red-green anymore. So just hopefully you'll never see a figure that expects the reader to distinguish between red and green. We've learned that we don't want to exclude a large portion of our audience who are red-green colorblind. At any rate, more complicated clustering, we can zoom in and do all sorts of clustering. And of course, now we use different color schemes to do that. But this data is really hard to navigate. If you look at it, this is JTreeView, which is one of the more commonly used packages. It's really hard to navigate through this data and see the relationships. And now we use the network view. This is a clustered network where we can see the interactions between the genes in this case. We can color them by different states. And it gives us a much cleaner representation of this data that we can explore. This happens to be a clustered network. And we can segregate between upregulated and downregulated and really explore this data in new and different ways. The same thing for single-cell transcriptomics, which I'm not going to go through in detail, but if you think about it, at the end of the day, single-cell transcriptomics wind up with cell-cell interaction networks and gene regulatory network inference. At the end of this long single-cell RNA-seq cycle, we wind up back with networks. And again, I don't want to go into too much detail on the single cell, but you can see how this could be useful. This, by the way, is taking advantage of doing some enrichment analysis within the scope of the network. Similarly for proteomics, I had some collaborators that came to me with this spreadsheet. They were very interested in looking at doing an analysis of skin treated with schistosomes. They were trying to figure out why some schistosomes cause schistosomiasis and others are much less severe. And this is their analysis. And you can see that they're looking at different substrates between the different species. I did a quick network analysis of this. And quickly you can see these are the shared and these are the independent. A much easier visualization. I could have colored it differently, but this gives you an idea on the kinds of ways that you can visualize very complex data. So if you think about networks, there's a number of different kinds of networks. Now, you've already heard Tina talk about pathways, metabolic pathways, signaling pathways. The key thing to keep in mind when you're looking at most of these pathways is they're typically pruned and they're typically curated. So you're looking at a very distinct, if you look at Wiki Pathways, for example, even in the COVID-19 map, there is a pathway that looks at one particular process. And I think tomorrow we're going to look at how to merge some of those and look at linkages between them. But typically, if you had all of that mapped into a single pathway, it would be way too complex. But they provide important mechanistic and contextual details. So my favorite sort of representation of this is this. Anybody recognize what this is? All right, this is where everybody has to wake up.
Oh, come on. I know the London tube map.
Perfect. The London tube map. All right, so quickly, is this the way you want to use to navigate above ground in London? The answer is obviously not, right? The Thames does not have right angle curves. I've walked down the Thames before. It does not have right angle curves like this. It is a diagram, a representation that's been curated to represent a very complicated process, which is getting around London. Think of a pathway in the same way. So you can think of the tube map as a pathway. But that's not the only kind of network that we have. Interaction networks are the ones that, you well-known protein-protein interactions. Here is a canonical yeast hairball, which used to be what people published. We don't publish these anymore because they're somewhat content-free. But you can also have protein-ligand interactions. You can have domain-domain interactions. And you can even have residue-residue interactions. For those of you who are interested in structural biology, you can see here is a protein shown in UCSF Chimera, and here's the residue-residue interaction network shown in Cytoscape. And so you can get different ways of looking at all of these networks. Here's one of my favorite examples. Again, asking everybody to wake up. Does this look familiar? Anybody know what this is? Oh, come on. The Olympics are starting. Everybody should recognize this.
Soccer, basketball, football. Yeah, whatever you want to call it.
Exactly. It's a football pitch. And somebody used Cytoscape to analyze two teams. And they were looking at how the passing density in terms of the thickness of the edges and the betweenness score. And you can see here, and we'll talk a little bit about betweenness, but you can see here that in this case, this team, everybody went through this person, right? So the right thing to do, of course, is you just sort of surround that person and then this team falls apart. So a lot of really interesting network analysis that can go into what might seem like mundane activities. The other kind of network that I spent sort of my first Cytoscape explorations with are similarity networks. The most canonical one is sequence similarity networks. And that's what this is. This is a sequence similarity network for the imidohydrolase superfamily. And these turned out to be extremely useful. If you look at this network down here, this particular one down here, you can see anytime you see gray, or actually let's look at this one. Anytime you see gray, that is a protein of unknown function, okay? Anytime you see a color, it means we know the function of that protein. Now, just based upon what I see here, I could very easily transfer the function to these gray proteins. There's nothing else confusing that. But if I look up here at maybe the gray proteins sort of in this in-between space between these two clusters of proteins, I cannot. I need to do more analysis in order to make some decisions and some functional associations about these. So protein similarity networks have proven to be very, very effective at helping us understand functional transfer. Chemical similarity, Tanimoto coefficients, people have probably heard about those. And I think that that's a very common network type. Ligand similarity, another kind of network. And in the non-biological world, you can just think of topic maps and tag clouds. All of these are looking at some sort of metric of similarity. All right, so that is the briefest of introductions to network biology, and we can go in a lot more depth to a lot of that. But what I want to talk about briefly is finding network data. Is how do you find the network? Now, first question is, what are the sources of network data? I want to be careful here. I'm not going to go through these, but if you're interested, you can dive through and find out where network data typically comes from at the experimental level, high throughput, low throughput, some of the computational techniques, and some of the public repositories. It's important to understand that, I will assert, because different experimental techniques have different strengths and weaknesses. And I talk about some of those in these slide sets. But in order to make sure that we can keep things moving, I'm going to skip that for now. But I would encourage you to go through and make sure that a lot of these techniques are at least understandable to you at the level that we're going to provide. But the question that I see a lot is somebody will come and send email to BioStars or the Cytoscape help desk and say, I have my transcriptomic data, and how do I turn it into a network? Or how do I find the network? Where is the network that is associated with this transcriptomic data? And the answer really is there's no such thing. There is no the network. It really depends on what you're looking for. There are hundreds of different interaction databases that will have different focuses. Biopsych, for example, if you're really interested in metabolomics, PDB, if you're looking at structural protein-protein interactions, if you're only interested in known complexes, you can go through this list and come up with different, here's Wiki Pathways for pathway data. But at the end of the day, it's going to depend on your biological question. And what is your analysis plan? What are you trying to show? You have to know what you're getting. So know about these repositories. And so if you're going to start with a gene list, you can do two things. I can construct a network from that gene list, and I'll show you how to do that, or at least one approach. There are several. Or you can find pathways that are enriched in these various genes. So what is it that you're trying to do? Recognizing that if you have a long gene list, you're not going to find, you're highly unlikely, I should say, in finding a pathway that has all of these genes in it. But you can certainly construct a network of protein-protein interactions that has all of those genes in it. Whether they're connected or not is a separate issue. So if we think about networks, there's a number of different approaches that we can use in Cytoscape to get access to networks. We can use this built-in app called Cyquick. There's Index, and that was on the slide that you saw Tina show earlier. And Index is, the best way to think of Index is it's like a Dropbox for networks. You can create your own network. You can get an account on Index. They're free, and you can store your network in Index, and others can share it. IntAct from the EBI, there's now an IntAct app, and you can download data from IntAct and present that in Cytoscape, or you can use String. And I'm going to jump to the string app and talk a little bit about that. I think it's one of the most general approaches. The string app has a number of different ways to query the string database. You can query by disease. So for example, you can ask for all of the genes, their proteins that are known to be part of Alzheimer's, and you can get a network of that. You can query by proteins. I can give it a list of proteins and it's going to download the data, and it'll give you a lot of additional data. Or I can do compounds. So if you are interested in metabolomics, you can sometimes get some interesting compounds from string where you're going to look at protein compound interactions. As with Wiki Pathways, Gene Mania is another similar type of thing. String shows up in this network search panel. You just select the kind of query you want. You can change species. You can change confidence cutoffs. You can add extra data as you're going through. And this is what you get back. You get a network that looks very much like what you would see on the string website. But you also get a lot of additional information. You get this kind of information, but you also get tissue-specific information, compartment-specific information, and you can begin tuning this. There's various sliders that you can go through and use. In addition to that, you can change confidence, you can expand the network, add additional things. You've got all these sliders that you can use to adjust what you're seeing, both in terms of the individual nodes, but also in terms of the edges and the confidence, the types of content, the types of edges that you're seeing. And string also has an enrichment. So you can do your functional enrichment right within string. It includes Reactome and Keg. So the question about Keg Pathways, you can enrich your network based upon Keg Pathways. Again, be careful of your background. That's really important. String will allow you to choose whether you want to use a whole genome background or whether you want to use another network as a background. So one of the things you can do is if you have a proteomics data set, you can take and you can bring down a network that represents everything in that proteomics data set, cluster that and subset it, and then you can do enrichment, but you can use your original network, that original data set that you had to do the enrichment as a background. So you can tune your background accordingly. So the other types of data, the Wiki Pathways app is one that you're going to use quite a bit. Just Tina's already talked a lot about this, but within Cytoscape, use this network search tool. You click on Wiki Pathways, you type in the pathway you want. You're going to get the option to import it as a pathway or as a network. And if you import it as a pathway, this is what you see. It looks a lot like the pathway that you would see on the website. And each of these are nodes in Cytoscape. And when you want to map to any data to those nodes, you can do that. And we'll show you how to do that in a little bit. You can also bring it in as a network. And this is a great figure in my mind to show clearly the difference between viewing some of these pathways as a network or viewing them as a pathway. You can see all this great information that we're losing when we view it as a network. Now, you can do network analysis when you view this as a network, but keep in mind that doing a network analysis like a betweenness centrality analysis on a pathway is probably not going to get you what you expect. In fact, this is a node here. So your betweenness centrality is not going to be a true, give you an accurate representation of betweenness of the genes. There's a number of different pathway databases out there that you can explore And this gives you a little bit an idea on something that I'm going to come back to with this, the plethora of Cytoscape apps. Okay, that was sort of high speed. Any questions about sort of that overview before we dive in a little bit to analysis and a lot into visualization? We okay?
Scooter, I have a question about the background. How can I set the background in the string app?
So what you do when you in String App, when you do the enrichment and you bring up the enrichment, there's a way you can either choose, there's a button where you say whole genome, or you can select another network and it will use the genes in that network or the proteins in that network as your background. So it's built into the String App, and when you run your enrichment, you have that option of choosing another network for your background as opposed to whole genome.
If I have a list of genes, I need to use them as a background. Can I do that?
Yeah, so what you'll do is you'll use that full list of genes,
Okay.
and you'll create a network from that, right? And that'll bring down all of the data for that entire list of genes. And then if you take a subset of that and you want to use that whole gene list for your background, you just point to that network that you created. String will allow you, the String App will allow you to bring down very, very large networks. In fact, if you're patient, there's a button that will allow you to load the whole genome for a species as a network. I don't recommend doing that, but you can.
Yeah, thank you. Sometimes when you put the names of the genes or the proteins, the Cytoscape, they didn't recognize your names. So yeah, so do you recommend any things like for example, any conversion tools like for IDs?
Well, ID Mapper and sort of, there's a number of different conversion tools. For string in particular, my recommendation, and actually I would say, if you're dealing with protein-protein interactions, I almost always suggest going to UniProt identifiers because they're pretty unique. And you often, pretty much all of the databases, unless it's a very, very new protein, will have all of those identifiers in them. So that would be my first shot at that. If you use the String App, when you put in your list of genes, if String doesn't recognize it, it will give you the option to, it'll give some guesses. And you have the option to look at those guesses and choose which one it is. You're going to be using the String App a little bit later. And so maybe you'll see some of that. There may be some options when you paste in a gene list where it's going to come back and say, which do you actually mean? So that's how String App handles it.
Thank you very much.
Sure. Okay, so let's talk about analysis of networks. These days, I would say primarily what people are looking at when they're analyzing these networks is I see a lot less detailed topological analysis and a lot more simple looking at node degree, which is has to do with the number of neighbors, a number of edges that come into a node or out of a node. And that seems to be sort of the focus, finding hubs and finding local modules. There's all sorts of other network tools and network analyses that I don't see used as much anymore. We used to be really, really concerned about whether this was scale free and a number of things like that, but I seldom see people doing those kinds of analyses on biological networks now. Mostly the three things that I want to sort of mention that have great biological sort of implications are degree, so the number of edges that a node has, You can think of that as a hub node. Nodes with high degree can be considered hub nodes. And if I look at this and think of this in terms of biology, this node participates, either all of these nodes are participating in a cluster, in a complex of some kind, or this node performs some critical function. And if I drop this node out, I'm going to have a significant perturbation of the biological process. So similarly, if I look at this blue node and think about the how this blue node, the role this blue node plays in this network, and I can measure this using something called Betweenness Functionality. And what you can see is, to get from this side of the network to this side of the network, everything has to flow through this node. So it has a very high betweenness centrality. And what happens if I drop this node out, you can see that the network falls apart. There's no connectivity between the nodes on this side and the nodes on this side if this node is perturbed or gets some sort of loss of function. Similarly, if I look at closeness, the closeness centrality, which roughly refers to how close this node is to every other node in the network, you can imagine a perturbation of this node impacting the rest of the network quickly, or more so than maybe this leaf node out here. And so an impact on this protein that might not be necessarily loss of function, but might be some sort of perturbation which causes enhanced binding or something like that, can quickly perturb the function of this network and hence the biological function. So that's sort of if you can get these sort of three concepts in terms of network analysis, that's a pretty good start. Those are the kinds of analyses that you'll probably wind up using. The other two analytical approaches that you'll see later, clustering is very common, and there's a number of different clustering techniques, but I could talk for hours on clustering and the relative clustering and dimensionality reduction approaches, and I'm going to hold myself back from doing that in the interest of time. And the other thing that we've already talked about briefly is enrichment. And saying, is this gene enriched in a set of terms vis-a-vis some sort of background? So feel free to go through these slides. If you go down, if you hit the down arrow, there's a number of different slides here that go into a lot more depth and give you the actual calculations for all of these, but I'm going to skip that. And instead, I want to talk about network visualization. This is Cytoscape's forte, and particularly from a standpoint of using this in an automation fashion, there are two things that you typically use Cytoscape for. Well, maybe, I'll leave it at two. And one is you'll use it for visualization. It has amazing visualization tools. And so even from R, you can take advantage of all this and you can use these visualization tools. The other thing is the plethora of apps, which have a number of different capabilities that are exported and available from within R. And I'll talk about that after our practical. Okay, so let's talk a little bit about depiction. So typically when you look at a biological network, what you really think of these days, particularly if you're looking at a protein-protein interaction network or something like that, is something that looks like this. This is actually in the Viz community. This is called a node link diagram. And you can see exactly where that name comes from. Nodes and the edges, what we would call edges, are the links in between them. But we can also split that up and look at a partition node link diagram where we're going to do some sort of clustering. In this case, we're using MCL clustering. We can use matrix representations. So this is a matrix representation, and you can see this highlights very nicely the local interactions. The global interactions are really lost in this. You can see that very light yellow splotches in sort of off link, but the local interactions, this gives us really good look at those clusters and how they group together. In fact, all three of these are the same network. So by using different depictions, all of these were done using Cytoscape, by the way. By using different depictions, we can begin exploring sort of different aspects of this network and focus on different connectivities, focus on different aspects of how this network is structured. There's other ways to do it. This is sort of an app called Gestalt, which gives a 2D representation. This is something called Biofabric, in which roughly speaking, these lines represent nodes. So it sort of flips the idea of node and edge on its ear. Great. So now I'm going to sort of focus on node link diagrams, because one of the reasons that I think node link diagrams are so useful is because we can take a node appropriately sized and we can map a number of different visual data onto that using visual mappings. So I can take data values and I can change the node fill color based upon those data values. I can change the border color based upon the data values, the border width, the size, the shape. I can change the opacity. I can change the label depending on what the data is. Similarly for edges, I can change the edge type, the color, the width, the ending type, whether it's a circle, whether it's an arrow. I can change the size of that, the color, all of these visual attributes I can adjust to take data that I want to map onto that. So for example, let's say I have transcriptomic data and proteomic data for the same network. And in terms of abundance, I could easily map my transcriptomic data on node fill color, and I can map my proteomics data, my protein abundance data onto border color. And I can see both of those. There are advanced ways where I can split the node fill color in half and use one side of the node fill for my transcriptomic data and the other side for my proteomic data. There's a number of different other ways that I can map this data. So the most common thing you'll see is continuous mapping. So I have some sort of numeric value, and what I'm going to do is map that numeric value onto, for example, size, or for example, the node color. And that'll be some sort of continuous mapping. I'll take a ramp or a gradient, and I'll find a way to take that node, that number that represents my data, and map it into that gradient. I can also use discrete. So I can say, if this is RNA, I want the node to be this color. If this is a protein, I want the shape to be this. If this is a kinase, I want the shape to be this. I can use any categorical data and map that discretely to shape, size, to basically any of these things up here. And then the last type is pass-through. So pass-through data typically used for labels, but there are other ways that you can use it. I will take that data and I will just map it directly. You can use it for color, for example, but you have to trust that you can map that color. So not all pass-through mappings. I can't say, for example, chartreuse as a color and expect that that's going to pass through, because Java doesn't understand what chartreuse is. So let's take a look at how this works. If I look at this network, okay, this is a interaction network that comes from one of the early papers that took advantage of the combination of protein-protein interactions and transcriptomics. And this is yeast data, in case you don't recognize the systematic terms of yeast. So the first thing I'm going to do is I'm going to change the node shape to just by default give me a little more recognition of the node shape so it stands out a bit. I'm going to change the labels to be the gene name rather than the systematic name. I'm going to change the edge type and edge color based upon the kind of interaction that it is. I'm going to change the size based upon the degree. And here's the mapping that I'm using for that. So it's a continuous mapping, and I'm setting up a ramp. So if it's degree 11, I want the node size to be 100. If it's degree 1, I want the node size to be 30. And that's how I got this. Now I'm going to use a color. And this is based upon my transcriptomic data. So I have some expression data, and I set up a nice ramp, and I've set up this color. And then I can add so a border, and again, color that border based upon a different transcriptomic data set. And in the end, you can take a look that I have this and the information associated with that. And we can go all the way back to that and begin to see how we can begin mapping data on and making these networks much, much more informative. Concerns. Avoid cluttering your visualization with too much data. There are dozens, literally dozens of different ways that you can change the visual attributes of these networks. And if I take advantage, if I have, let's say I have 50 columns and I try to come up with a way to map all those 50 columns in various visual attributes, you're going to completely confuse yourself. You're going to completely confuse any kind of viewer. If you have multiple ways of looking at this, consider creating multiple images. So this is, a brief foray into admitting my age. When I used to work for a pharmaceutical company called Genentech, and in the early days of Genentech, one of the vice presidents and researchers was really, really interested and loved Macintosh. So we got a whole bunch of Macintoshes and these with these little tiny, postage stamp size screens. And everybody was excited about Macintoshes for two reasons. One is they came with this cool program called MacWrite. And two, you could have laser printers. Before the Macintosh, printers that would do fonts were extraordinarily expensive and very difficult to use. But now with the laser writer, it's sort of got to be a desktop type tool. And for a while, MacWrite just has this incredible series of fonts that you could play with. And for a while, everybody was playing with those fonts, and you would see documents coming out of the laser writers that were just impossible to read. They had 5 fonts, they had different styles, they had all of this stuff coming up. And the net result of that was that it just looked like clutter rather than cute or rather than informative. Over time, people figured this out, and they occasionally, one, you don't even see the cigar store font anymore in Mac or anything else. And it's sort of narrowed down to, you chose a font family, you stuck within that font family, and then occasionally you would put in something for fun at the end or to highlight something. You need to do that same kind of thing with your networks. Don't clutter your networks with tons and tons and tons of different visual attributes. Really focus on what's important. And then just if you have to, if there's 2 different things that are important that you want to highlight, create multiple network images showing exactly the same network, but highlighting different things through different visual attributes. That can often be far more effective for the reader than merging them into a single one. Okay, layouts. So layouts determine the location of the nodes and sometimes the paths of the edges. Simple layout is grid. It's very, very fast, and it's probably marginally useless. But at least it gives you some idea on what your data looks like. The only way that I've seen this typically used is if for whatever reason you're starting with no edges at all. So if you just have data and you haven't yet, for example, I will see this sometimes when people bring in compounds into Cytoscape, and they'll have 10,000 compounds. And now what they want to do is create a similarity network based upon Tanimoto coefficients. So they'll have a network which doesn't have any edges to start with. Hierarchical, here's a great example of how not to use hierarchical layouts. This is not hierarchical data. So using a hierarchical layout here is only confusing, and it really obscures the data. So use hierarchical layouts when you have hierarchical data. Circular layouts, here you can see I've arranged this data in a circle, but I've also ordered it based upon degree. So I've sized the nodes and colored the nodes based upon degree. And you can see how ordering it this way gives me some visual idea on where my high degree and my low degree nodes are. What you typically see when you look at this is you typically see force-directed layouts. So a force-directed layout just simply, it simulates all of your edges as springs. And those springs could be weighted or unweighted by some value. And then you basically take all of this as springs and weights. And then you sort of let the physics work and settle out. And then you smash it down into 2 dimensions. And note that's really important. You're smashing this thing down into 2 dimensions. So what can easily happen as you're going through the simulation is people expect that the weights will always hold true in terms of actual length. So if I have 2 nodes here and 2 nodes over here, they'll expect that if this edge has the same weight as this edge, that these 2 edges will be exactly the same length. And that is very, very seldom the case. Due to this optimization that's going on to try and get it down to 2 dimensions. And here's an example of a force-directed layout, which is what you're commonly used to seeing. Note that you're not constrained to only using one layout. So I've seen a lot of very, very effective visual networks where people have used a single layout and for the overall network, and then taken a piece of the network and done a different layout within that piece of the network. So that can also be very effective. Multi-layer layouts, partition the graph and you lay out each partition. There's a number of different algorithms that do this. And there are many, many other kinds of layouts. Several apps exist that do layouts. So there's the whole suite of Y-Files layout apps that you can download. There's an app called Boundary Layout that I wrote with a student. And what it does is it allows you to constrain nodes within a boundary. And then it does a force-directed layout within that boundary. So for example, you can imagine drawing using annotations. You could draw something that looks like a cell with cell partitions. And then based upon the partition, you can lay that out within there. And that's called boundary layout if you want to explore that. Now, you're not done when you look at this, you do your layout, because sometimes it just isn't quite what you want to show. So there's a number of manual tweaks that you can make with Cytoscape using these node layout tools. I can select a group of nodes and I can scale them, so I can push them apart or bring them closer together. Often what I find that I do a lot is I'll take a look at the force-directed layout, and there'll be a part of the network that overlaps. So let's say that in my force-directed layout, I have something that has like, let's say it has 5 nodes out there that look like this. And for whatever reason, that overlaps another group of nodes. I'll just select those 5 nodes and rotate them so I can get a clearer layout. These node layout tools are incredibly useful. And most of us that use Cytoscape a lot use them quite a bit. So I would encourage you to play with those. So layouts, use layouts to convey the relationships between the nodes. That's what they're for. What you really want to do is highlight if I have 2 nodes and they are closely related in some way, I want the layout to show those nodes are closely related. You can do that a couple of different ways. The layout can put them close together, and you can also highlight that by maybe making the edge thicker or darker, depending on things. There's no such things as one correct layout. You may want to try different things. And in particular, force-directed layouts need to be tuned. So you can bring up the layout settings panel, and then once the layout settings panel is up, you can go through and you can set an edge column to use for a weight. I can set the number of iterations that I want this to do. I can set the coefficients, the default spring lengths. I can set node mass that'll speed up or slow things down, and sort of a number of different parameters. Each layout algorithm will have a different set of parameters. And again, I often will wind up playing with these to get the layout that I like. The last piece of this is animation. Animation can be extremely effective to show changes in a network or compare 2 conditions in a network. So here's an animation that was done using Cytoscape. And what's going on here are I have 2 sets of proteins, actually 1 set of proteins and 2 sets of edges. In one case, I'm clustering these proteins based upon their sequence similarity, purely based upon sequence similarity. In the other case, I'm clustering them based upon the similarity of their ligands. Okay, so if 2 proteins have very similar ligands, then I'm going to call those 2 proteins similar. So first principles, sequence means function, sequence defines the function defines, sequence defines the structure, structure defines the function. Therefore, these 2 networks should be almost the same. They're not even close to the same. So this was an animation that I was asked to do by one of the faculty at UCSF. I did this. I looked at it. I said, something is really wrong here. I showed it to him. He looked at it and he said, great, that makes perfect sense. And he walked away, leaving me to wonder what I had missed. So anybody see what might be going on here? Any guesses? You're looking at sequence similarity versus ligand similarity. Well,
sequence is a structure.
Right.
Yeah.
Well, and not only that, sequence similarity, ligand similarity is going to depend primarily on a very local sequence, the pocket, right? Whereas if I'm using something like BLAST to do my sequence similarity, it's going to be very broad. So I could have structural components of these proteins that have little or nothing to do with the binding. And it's the binding motif that's really important. And when I finally tracked down Brian and said, okay, this is what I think is going on, is that I have a global similarity measure versus a local similarity measure. He said, exactly right. That's what I expected to see. So It was a fun exploration, and I've used this multiple times now to show this. By the way, if you want to make your own animations within Cytoscape, there is an app for that. It's a very simple app called SciAnimator, and it allows you to basically take keyframes of different networks and then of a network as you change it, and then animate across it. Okay. So that's analysis and visualization. So how are we doing? Questions about any of that before we sort of actually tie all this together in Cytoscape?
That is wonderful, Scatter. I have one question regarding that. Okay, so you have a network, you have some hub genes, you have some like you measure all the degree and other things. So my question for you, because you have much experience, like how can you translate this network to something like biomarker discovery, some drug targeting, or like for example, okay, I have a network and I have some nodes, I have some connection, but how can I choose one of them to further analysis, or what shall I do with this network? Like clinician, they ask, okay, you have a wonderful network, so what else? What's next?
Good question. And I think you're going to be doing a lot of this, the what next, as we go through the next 2 days. But the short answer is it depends on the biology. So if I have typically, let's take a transcriptomics experiment or even a proteomics experiment, where what I'm seeing in my network, okay, I'm going to download, let's say I'm going to use strings. So I'm going to download the known protein-protein associations. And then what I see based upon protein abundance is that this node in this particular condition that I'm interested in has a substantially lower abundance than I guess you could consider it the non-disease condition or non-experimental condition. And now what I would want to do is I would want to first take a look at the enrichment. I would do some sort of functional enrichment to say, this node is involved in this process. Or I can take a look at all of the nodes that are substantially downregulated and say, what processes do I see that are downregulated in this condition? And once I've done that, now I can begin looking at these particular genes, these particular proteins, and start asking different questions. And the whole idea, I think, with a lot of this, if you look at network analysis, there are two really important functions here. One is to show your biological results. So I already know what's important. I've done the experiments, I've explored my data. What's really critical is that this particular protein is substantially downregulated when in this particular perturbation, whatever that perturbation might be. And I want to show how this leads to some phenotype, for example, okay? And then I'll show a network that shows that in this particular case, this pathway, this particular protein is downregulated in this pathway. And then based upon that, I can show based upon this pathway that it's leading to this phenotype, okay? The other thing that I'll use networks for is exploratory analysis. I don't know what it does. And now what I'm trying to do is develop a hypothesis based upon looking at enrichment, looking at interactions, looking at proteomics data or transcriptomic data. And then I want to go and verify that hypothesis in the lab. So almost always, when you're using these tools as exploratory tools, particularly for some of these large scale experiments, at the end of the day, what you're looking at is formulating a hypothesis that can be experimentally verified. So even in the COVID-19 maps that you're going to be spending a lot of time looking at, a lot of this data, you're going to come back to saying, okay, here's a hypothesis at how this impacts disease. And now I'm going to go in the lab and I'm going to see if that's the case. So sort of two different ways to look at this.
That's great. So can I like compare two networks? For example, when I knock out one of the nodes, like then I say, look at this. When I knock out this node, look at this, the network change completely. So then I can just, I don't know, can I do that?
There are a number of different network alignment tools. The area that you're sort of talking about is network alignment. Cytoscape has the ability to merge networks. And when you use the merge networks, you can get a difference network. So it's not really that helpful. If you think about it, if I just drop a node, right, you can visually, you're not going to see the network rewire unless you have, unless your data set is actually a APMS data set that your data is coming up with the networks. There's a great paper on network rewiring between Cerevisier and Pombey that was published by Trey Idecker and Nevin Krogan that sort of shows ways that you might do that. But their data was the network. If you're downloading data from string and you knock out a node, you're just going to get that same network without that node and without all the edges that are going to that node. And it may not be as informative as you think it might be. Now, the other way to do that is if you have a data set of transcriptomic data, for example, and you see that this particular protein is overexpressed or significantly abundant, you can use a diffusion map. A technique called diffusion, heat diffusion, which will show the area of potential impact. And then you can take that diffusion map and this is taking advantage of a technique called guilt by association. And then once you do that, you can sort of do the enrichment and say, these kinds of things are being affected. So that's probably a better approach.
That is wonderful. Thank you.
Sure. All right, so let's dive into this thing called Cytoscape. A couple things to note about Cytoscape. It is open source. If anybody doesn't like the way part of it works, you're welcome to download it and fix it and give us a pull request. We would love to see that. We would be happy to acknowledge that. It's cross-platform. It's sort of part of our bane and our boon. It's great that it's cross-platform, but there's just enough platform differences as that we as programmers have to deal with that can sometimes provide a bit of a challenge. And it's developed by a consortium. So UC San Diego is where the home is, but Institut Pasteur is part of the consortium. ISB up in Seattle is part of the consortium. UCSF, Gladstone, University of Toronto, all part of the consortium that work together to develop this platform. Cytoscape has two critical concepts, and it's very important to keep these separate in your mind. Networks, which are nothing more than nodes and edges. If you think to the node link diagram, that's all it is. So strip away any biology, strip away any additional information you have, transcriptomic data or anything else, even node names, strip away all of that. And it's just an internal representation of a graph, okay? Then there's all these tables, and tables provide a lot of different information that we will link back to an individual node or an individual edge. And the magic of Cytoscape is these things called visual styles that allow you to take data in these columns, in these table columns, and map it to the visual attributes on nodes and edges. And the fact that Cytoscape, in its original, way back from the start, kept these concepts very, very separate, has allowed Cytoscape to be used for a number of things completely unrelated to biology, like how a soccer team does their passing. Cytoscape's also been used by a science fiction author to keep track of his star maps for his stories. Or it's been used for water distribution systems in cities. All of these are very far afield of what most of us think of Cytoscape, which is a biological network visualization and analysis tool. There is no biology in the core of Cytoscape. All of the biology comes from apps, including some of the core apps and also apps provided by others. And leading right into apps. So there's an app store for Cytoscape. I don't like the name App Store. I debated against the name App Store and I lost that debate. We don't sell anything, and so it's not really a store. It's an app repository. And you can go there and you can find apps that are of interest to you, such as the Wiki Pathways app. And you can download that app. Or if you have Cytoscape running, you should be able to directly load that app into Cytoscape. There are currently over 300 apps. It continues to grow, and new apps are showing up, all the time. It's slowing down, I would say, but there's still a great interest in exploring new apps. I'm on a project right now which is building a huge graph database. Contains everything from food to chemicals to genes to proteins to pathways to diseases, phenotypes, symptoms. Trying to think of what else is in here in Spoke. And it's obviously web-based and people can browse it. But now there's an increasing push to get a Cytoscape app that will allow you to query this and get the neighborhood within Cytoscape. You can also find apps directly from within Cytoscape if you don't want to use the web. And you just, this just gives you a list of apps. And if you click on an app, you can get a short summary of that app. You can load network data from Excel, TSV files, CSV files, XGML, SBML, BioPax, PSIMI, CIF, GML, and you can load apps such as the Wiki Pathways app, which will allow you to import different kinds of network data. And similarly, you can import data from all these repositories. And we're going to talk a lot more about automation, so I'm going to skip that for now. Now, remember, that was network data. You can also load table data from Excel, tab separated files, CSVs, all that kind of stuff. And in fact, you're going to be doing that very soon in the hands-on exercises. In Cytoscape, when you do a save, you're saving a session file. So when you open, you do an open, you're opening a session file, and a session file just contains everything. It contains screen sizes, styles, tables, networks. In some apps that I've worked on, I actually write PDB files into the session file so I can remember it. Images will go in the session file, depends on really the app that you're working on. I worked on a single cell RNA-seq analysis app in Cytoscape, and we saved the single cell data in the session file so that you don't have to reload it. All sorts of different approaches. And of course, you can get publication quality graphics in a number of different formats. PDF is obviously my favorite because it's nicely scalable, but you can also use SVG. And if you're going to use PNG, I strongly recommend that you use at least a 300% or a 500% sizing of that. So when it converts it to the image, you get a much denser image. Okay. A little bit about Cytoscape. I think we'll just jump right into the hands-on exercises. How are we doing on time? Not bad. So here's the exercise. If you go to the first link that was out there, you can click on that. And what you're going to do is you're going to do a very traditional bulk RNA-seq analysis using Cytoscape. And this should help you go through, let's see. Oh, thank you, Lauren, on top of it. So if you go there, you should be able to link on the practical. I don't know, Lauren, are we going to use breakout rooms for this, or do we want to, since we're a relatively small group, should we just keep it right here? I don't.
So I think maybe for this one, if we keep it here, because my breakout rooms are not working currently.
Well, that's a good reason to keep it here.
Yeah.
Okay. All right. So basically the rules of this game are just go through. Hopefully everybody has Cytoscape already installed. You're going to download some data as an Excel spreadsheet. That's the way most of these analyses start with Excel. Once you've done that, you're going to do some sorting and some filtering, and then you're going to basically use the string app. And here's the question on how to clarify. Here's an example gene that wasn't found by string, and it's giving you the option of importing and clarifying that. You're going to get a network, and then you're going to go through and you're going to do a standard analysis of that. You're going to bring in that data set and you're going to do the visualization. And at the end, you're going to wind up with a network that looks like this, where you're going to have enrichment as a donut charts around the outside, and you're going to have the transcriptomic data shown as a color on the nodes. Okay, that is a very quick overview on what's coming. And with that, I'm going to let everybody get at it. Good. Anybody have any questions about the practical or any of the general Cytoscape background before we launch into automation? So avoiding the question in chat was how to avoid overlap of nodes. And this is where, as I said, going in and tuning the layout algorithm will help. The boundary layout app actually has a mechanism where it tries to push nodes apart so that they don't overlap. So you can try that. The other layout algorithm that's often has really aesthetic results is the organic layout. The problem with organic is it's not edge weighted. So if you want to do any edge weighting or do any tuning, you can't use organic, but it's often a very, very solid layout algorithm. So, oh, okay, there's a new remove overlaps in Y files. They update every so often, so I'm not up to date on that. Thanks, Tina. That's news to me. That's great. That allows you to use something like Perfuse and then use that to remove overlaps, which is a great way to do it. Okay. Wow. I learned something new either about science or sometimes even about Cytoscape as I give these tutorials. Okay, so let's dive into automation. The first thing that we want to do here is get a brief overview of, yeah, I'm still me, by the way, I haven't changed. A brief overview of how we got here. Back in the Cytoscape 2 days, which was a complete old architecture, I found myself doing a lot of essentially repetitive tasks in Cytoscape. And so I implemented a real quick and dirty command line processor that you could have a bunch of commands in a file and it would go through and it would execute those commands. And that got brought over into Cytoscape 3, enhanced a little bit and fixed. At various points in times, we tried to make it into an even more elaborate scripting language with loops and parameters, and quickly realized that that was just the wrong approach entirely. And what we did instead is we implemented something called scirest. And all scirest does is it has, when Cytoscape launches now, it opens up basically a web server, an HTTP server that opens up as part of Cytoscape. And it listens on port 1234 by default. You can change that. And it accepts REST, representational state transfer, commands that can access the internal data model or ask Cytoscape to perform various functions. And notice that scirest also uses the same commands that were already implemented. And so you can access these commands via scirest, and you can also access various information about nodes and edges and other things via scirest. So this was really, really useful, and has all sorts of capabilities, but at the end of the day, it's really low level. And it wasn't really tractable for a lot of people to take advantage of. So what happened was there was an implementation of R called RSci3, which took advantage of scirest and provided a much higher level library that allows you to take advantage of both commands and REST and to perform automated functions and utilize Cytoscape. More recently, Pi for Cytoscape is the Python equivalent of RSci3. They have very, very similar functionality, and there's a lot of effort that goes on in the two teams to make sure that they are in sync. Why would you want to do a script? I don't think I have to, really sell this audience on that. If you want to do things in loops, if you want to be able to repeat it in the future, if you want to share it with colleagues and say, hey, give this a try, or if you already have an R or a Python pipeline, and what you really want to do is you want to export that data into Cytoscape, save it as a session, and then send that session off to a collaborator. So there's a number of different automation examples out there. There's some sites that you can go to that will give you a number of examples of using Cytoscape in different ways. I would recommend that if you really want to do something, go see if somebody else has already done it. And I think, so for example, this came up just recently. I don't know of anybody that's developed a Cytoscape notebook that takes advantage of Cyanimator and automatically creates a Cytoscape animation. So that might be a useful thing to see up there for others to take advantage of. This gives you an idea of the breadth of the commands that you have in R. This is from the R package from RSci3. This is old. It's much, much more expansive now to give you an idea, but I couldn't find a slide where I could fit that all into one slide. This is from the 2-year-old paper that was published on F1000. And it's even easier to use now, particularly the commands capability where you can quickly just do a commands run and get the results back. And there's a link to the full site RSci3 reference, which you can go off to and see how you could do this. And there's the cool thing is a number of apps basically export either REST interfaces themselves or commands, as the command syntax shows. And so a lot of the apps that you'll find in Cytoscape are accessible to you from R or from Python. So for example, as you'll see in the use case that we're going to go through, you can have R basically ask the string app in Cytoscape to load a network into Cytoscape. By sending commands to string and say, load this network with the taking in advantage of these proteins. So this provides a lot of capability and opens up a wealth of apps to you without having to recode a lot of that stuff. And at the same time, you can take advantage of a lot of the capabilities that are within R and the R packages that are available. Okay, we've already talked about this. So here's what I'm going to suggest we do briefly is I want to get through this setup. So what I'm going to suggest we do is, let me see if I can get out of full screen mode here. Come on. There we go. I'm going to stop sharing this. And I'm going to share, at this point, I have to share my entire desktop. So there's going to be this weird flick. Okay. So everybody probably has something that looks like this in their Cytoscape. If you've already exited Cytoscape, go ahead and start it up again. And then in Cytoscape, come up and do a file close. Now, what this is going to do is it's just going to wipe everything clear, and it's going to give us a clean slate. Great. All right. Now then, I have our studio running. So if you haven't launched our studio, go ahead and do so. And what we're going to do is we're going to just take and make sure that we're set by, I'm going to copy and paste into our studio. And that's it. Okay. So what this will do, this may take a little bit longer if you haven't already done this. What it will do is it will install RSci3 and make sure that it's done. If Cytoscape is running and RSci3 is currently loaded, you should be able to copy the Cytoscape ping and say, hello. And now we're talking to Cytoscape. So if hopefully everybody has gotten, I'm going to give us a couple of minutes to get there. And what we really want to do is get to the stage where you are connected to Cytoscape is available on your in our studio. So how are we doing? Can I show a sign of, see a sign of hands? Has everybody got that far where you are connected to Cytoscape? Great. Yep. Still looking for thumbs up. I got 2 thumbs up so far. Are other people running into trouble, or are you not back from break? RStudio and Zoom are a bad combo. Interesting. Okay. That's going to make a lot of, you should be able to do the same thing just in R. RStudio just happens to be very convenient, but you should just be able to launch R and type in the same commands. There's nothing RStudio specific. Tina, do you agree? Am I off base on that? Not being I'm more of a Python person than an R person, so.
Yeah, you should be able to, I guess, also just do it from R. For tomorrow, it's a little bit easier in RStudio because we have the R markdown files, but then we'll work in breakout rooms, so it might be different. I don't know.
Okay. I see. So this is the first time that you've loaded RSci3. Yeah, it'll take a while. Because brings in, you've got to bring in a number of different packages. So the next section isn't hands-on. So the next section, I'm just going to talk about a couple of things. Felicia, does it work for you? Are you able to get it working just slowly? Great. Okay. So the next section here is just, I'm going to talk about some approaches and how to go about thinking about doing your automation. And so we're a couple minutes away from where you have to get to this, you are connected to Cytoscape. So just keep plugging away on it, and then let's see how we go when we get moving again. Okay, are my slides showing now exclusively, I hope? Great. Okay, so in reading through this, I looked at some of the previous discussions about how to go through and approach some of your automation steps. I mentioned apps and automation. Be aware that as part of this web interface that Cytoscape provides, it also provides something really cool called Swagger. And with the Swagger, if you go to Help Automation Cyrus command, and those of you who are still working on your install, don't do that now. let that complete. But you'll wind up with this page. And what this page gets you is for each command in Cytoscape, for each Cyrest interface, detailed description. So this is for the string protein query command. And you can see that it's going to tell you exactly what it's going to do. It gives you detailed descriptions of all of the parameters that you can include. And you can go through this list and decide exactly what parameters you want to use. And it'll also give you the opportunity to type in and try it out, which is the thing that I really like. You can type in what you want, what the parameters are and the values, and then you can just say, give it a try, and it will go off and it will try it, and you'll get the results back, or you'll get an error. And that can sometimes be a lot easier, or at least I find it easier than being in RStudio and trying it and then getting something back and not knowing what's going on, because I miss all of this detailed documentation. There are ways in RSci3. There's the commands help that will give you an abbreviated help. But in general, I think the Swagger documentation is much, much more complete. So you can look at all namespaces and commands. You can get more detailed documentation. You can test things, and you can look at the results. So in thinking through this, and one of the things that I like to really think through is, how do you go about, if you're going to automate something, how do you go about it? So my recommendation in particular, unless you're really an R guru and you just kind of R flies from your fingers, I always would recommend that you do the part of the workflow that's going to use Cytoscape using the GUI. just sort of step through it in the GUI until you get what you want. That visual graphical user interface allows you to kind of make sure that your each step is exactly the way that you want it. It's much easier to back up and redo it in that context. Determine which commands you want to use. You're going to use Swagger to do that. And then when you get back to R or Python, you can use the commands run to just stick those commands right in there and have that executed directly in Cytoscape. Okay, so questions. Does that all make sense?
So my understanding that if you need to do like some commands, you just write it in R, then this command go to the Cytoscape, then you retrieve the result from Cytoscape to R again, or is this correct? No.
My suggestion is actually just the opposite. My suggestion would be to start in Cytoscape itself. So if I go to Cytoscape, okay, using either the Swagger documentation or you see this little button down here, I can bring this up. And so, for example, there was a question earlier about whether Cyanimator, I could have Cyanimator commands. And so I just did a help Cyanimator. To see all of the namespaces, I can just say help in this little box. And these are all of the namespaces I have. And you can see a lot of these like enrichment map, cluster and cluster dim reduce, cluster rank and cluster viz. These are all part of Cluster Maker. Cyanimator. So for example, I could do a help cluster. And you're going to see these are all of the algorithms, the cluster algorithms that are available in Cluster Maker. And I can get more detailed information by help cluster MCL. And now I can get all of the information about those commands. Now, in general, this is often not as much information as you'll get from the Swagger documentation. But it does give you an idea on what kinds of things you can do. And I can just type in the command that I want to execute right here and see what happens and do I get it right. And then if I didn't, just close it and then try it again until I get it right. Then in R, I can take that exact command string and paste it into commands run in R. And that allows me to just do this quick prototyping here. And then when I've got the workflow that I want, the various commands that I want, I can go back to R and I can just string them all together and then get the results back at the same time. Does that make sense?
Yeah, so what kind of language in Cytoscape? Like it is, they have different language or?
It's all the same kind. It's all this very, very simple cluster MCL argument equals value. Everything is that simple.
Gotcha. So for example, give me like one example, like for example, cluster, what cluster for what? Like how can I, like, can I pass a network or a list of genes or like?
Typically a network. So if I have a network open, I can say cluster. Well, let's see, I can bring open a network. Now this is just one command. There are many, many different commands. But if I, to move this out of the way, go to the home screen, open this, and I can say cluster MCL network equals current. And I can say cluster attribute equals, oh no, sorry. Attribute equals, I need an edge value, edge betweenness. Okay. And that's going to go and that's it. It did the clusters. And now if I look at the nodes, I scroll all the way out. Here is the cluster designation for each node. Okay, and there's a command to create a network, but again, without going too far into clusters, the whole idea here is I can just execute these commands from R and have the right thing happen in Cytoscape. So, okay, good. So now hopefully everybody has our Sci3 loaded. Now we're going to go through this use case. And again, this is sort of hands-on. And we have until, so we have 30 minutes, roughly. Can I steal a couple extra minutes, Lauren? We have 30 minutes to go through this. And most of this is going to be, once you have our Sci3 loaded, it's going to be just that same thing, whether you're pasting into R or pasting into our studio. Here are the commands that you're going to try and paste in, and just go through. You're going to download two different data sets, an expression matrix and a gene ranks file. And then you're going to go through, this is just background data. And this is, you're going to type, add this in, this is what you'll see. And then you're going to go through and you're going to basically type this into string, and you're going to get data back from it. And you're going to be able to create a network, get the network back, do screenshots, and basically explore this network from within R. And the net result is, once you've done all this, you will have a whole list of these commands. They'll be in R or R Studio. You can make a notebook out of it, and you can repeat this process whenever you want, possibly for different genes. Okay. So with that, I will turn you loose. And I'll be probably working through this myself. If you have something already up in Cytoscape, I strongly recommend that you clear that first, that you do a close. Like that. So just in case somebody else hits the same thing, I'm getting stuck on the set node shape bypass. There seems to be a bug in here. So it's not critical. I would just sort of skip it, but just be aware. If you get past that, great. Is anybody else seeing the same error that Fadi is? I got through that one without a lot of.
So what Scatter, what I did, I just download them myself, then I read them using read.csv. So this is how.
Perfect.
How can I get? You know?
Okay. I'm not sure why that failed for you. I had other things fail for me, but that one worked. By the way, if anybody sees this error, this commands file transfer error, it's getting ready for Cytoscape 3.9, which will have a new app called the file transfer app. So it works just fine. It fails back to the non-file transfer version. And it turns out, even though I was getting that error about the bypass, it was working. So I don't know what was going on there, but it did exactly what it should have done. Philip and Felicia, were you guys able to get past your first step? Are things working for you now? So my recommendation would be, because you're going to need this probably tomorrow. So just get through all of that update after and or let it keep grinding. Yeah. And then you may want to run through this step by step. It's pretty quick. And just make sure you have some of the concepts. Yes, I see you nodding. Yeah. I did that. See, I knew this was coming. I did all that last night.
I get like problem like try to connect to 123 the local host port.
Mhm.
So this connection refused. Is it because of the this is from the RCY package or from
You're trying to connect to that using curl or using just this should just work. The layout network, right? If I just go here and just paste that in, right? I shouldn't get an error. But that doesn't involve curl at all. So I'm wondering if that error is just left over from the curl command that you did.
yes, so the curl is not working in my machine. I don't know. Maybe it is one of the dependency. I need to upgrade it. So you know, like when I install RCY package, they said you have to upgrade these package. Then I just say no.
Oh, okay. Yeah, I would definitely say yes.
Yeah, but if I upgrade it, it will like crash other things I'm doing. Like the I'm trying to do like some I'm using only specific version of the package. I cannot upgrade them. So I
Okay.
think the best way to do it, I think if I can either two-way. First, we can like work in RStudio cloud or just using Docker or something else. So you cannot like um, like do something and crash your RStudio. Mm.
Yeah, I see the problem. It's difficult to do this in the cloud because obviously Cytoscape has to be local.
Got you.
And I don't know why my import 1234 is open. You can open that up in your web browser. So I'm not sure why you're getting a connection refused. Is Cytoscape running?
No, I close it.
Oh, well, that's why you're getting that connection refused.
I got you. I thought you said you have to close it last time. Like,
oh, it's closed. There's a close command in Cytoscape. Sorry. I understand now why that was confusing. If you go to File, Close, that will just close the session.
Got you.
That explains everything.
Thank you.
Sure. Okay, so yeah, yes, yep, yeah. Blame Alex for that, Tina. Close was his idea. Yeah, we should have, in that file, we probably should have a quit or something like that. I know there's already one here, but it'd probably be good to show here. That way it'd be clear that that's difference from close. Okay, so the goal here was to just give you an idea on the kind of things you can do with automation. Obviously, there's a lot more. And since most of the time you were just copying and pasting, my recommendation would be to try and take a close look at what you were pasting and think through exactly what that's doing and how it might impact or fit in with your particular workflow. Tomorrow, you're going to be doing a lot of the same thing, although I think you're going to have to do more fill in the blanks kind of things than just copy and pasting. So you're going to have to think through that. So I would definitely suggest going through this a little slower and think through what exactly is going on. And with that, I will turn it over.
Okay, thank you, Scooter. Okay, so I think basically now I only have a very short wrap up to do. So I will just share my screen and then we're going to go through just a reminder of what we've done today and then just a look ahead to tomorrow. Okay, so I think you should be able to see my my screen. So go to slideshow. Yeah. Okay, so what are we getting into today? So just to remind you, we did a brief introduction to the COVID-19 disease maps project and Wiki Pathways. And then Scooter gave an introduction to Cytoscape and to Cytoscape automation with R. So yeah, that's basically what we covered today. And tomorrow we're going to use all of this stuff to kind of get into some data analysis with the COVID-19 data set. So this is what the schedule looks like for tomorrow. So we start again at the same time. And Martina's going to introduce these workflows, and we're going to end up going through 3 of these workflows. And then at the end of the day tomorrow, there should be some time to get some input on your own project. So the data set that we're going to be providing, that we're going to be working with, is this data set from Geo. So the number there, if you wanted to look it up, it's from this paper. So the imbalanced host response to SARS-CoV-2 drives development of COVID-19. And basically, this data set comes from these normal human bronchial epithelial cells, NHBE cells. And then there's 3 samples that are infected and 3 that are mock treated. And these are, of course, then put through the RNA-seq, and we compare. And then we're going to run these workflows where we have this kind of automated Cytoscape and R analysis. So that's going to be applied in these 3 workflows. So we're going into first, it's a pathway enrichment and network visualization and drug target extension workflow. The second one is going to be protein-protein interaction networks and pathway information extension. And the final is the pathway crosstalk analysis. So I think that's it for today. Are there any questions or anything about what we're going to see tomorrow?
Yeah, Lauren, I have one question, 2 questions. First, can I have these slides you are just presenting? And the second question is, do we need to do anything before tomorrow, like installing something which is like fast the process instead of like, do you have some package to install or something like that?
Yes. So give me 1 second to grab. So this, I'm going to send in the chat right now. So from the page that I'm sending now into the chat, you can get all of the slides and the practicals from there. There's also on the top this instructions page, and I'll also put this in the chat. So this is the instructions page for the one that I've just put in the chat, and that's for the things that need to be installed before tomorrow or for tomorrow's practical sessions.
But I saw that the slides and the practical and the other things, there is no link for it for that tomorrow.
It'll be there. It'll be there. So it's, yeah, maybe not at the moment right now, but it will be. Yeah, you're right. So the intro to the workflows, and then the practicals, indeed. So we will update that before tomorrow, before our session.
Yeah, it
so
will
so it'll
like
be available.
Yeah, it will be, Lauren. It will speed up if we have it before. Then we can just go through them instead of like asking stupid question like.
No, that's fine. So I have them nearly ready. The thing is that I wanted to see what people want to get out of the workshop. So I made now during this workshop an update to workflow 3 and include some proteomics data there. And we'll try to do some animation. So we'll see if that works out. But I can already make the links. There might be slight changes to it tomorrow. That's the only thing that I said. I wanted to react to some of the requests that came up. But I can put everything there already.
I don't know if this is possible for tomorrow. I'm interested to look at the single cell things you just posted, like the
yeah, there's not so much single cell data for COVID that I now would have immediately access to. So I don't know, we will be able to, I can check if I can find something tomorrow, and then I'll see if I can integrate it as an optional workflow or something. I'll have a look at that.
Amazing. Thank you.
Great. Any other questions or comments or anything? No, then I think that's it for today. I don't know if people want to continue finishing up working on that, the practical parts, because we have a few minutes left. So I guess if you want to continue, please feel free. Otherwise, then if you're done and ready to head off, then thank you for the session today. It was very nice, and we'll see everybody tomorrow as well.